Okay, guys, let's go ahead and get started. So just to give you an idea where we left off last time, we were talking about DNA replication. And we talked about DNA polymerase, how that worked, how DNA polymerase can only work in one direction. So when you have this, the DNA sort of opens up, for one direction, there's really no problem. It just sort of moves along, adding the nucleotides. For the other direction, it sort of works in little spurts. It will lay down some nucleotides in a region that's called an Okazaki fragment, and then we have DNA ligase that ties these fragments together. Okay, so that's kind of where we ended last time. And now we're going to move on to the next topic. So we're going to look at gene expression. Some of this is probably familiar to you. So when we talk about gene expression, what we're talking about is the idea that we have genes and we're expressing them. And typically, we're talking about expressing them as proteins. Now, what I'm going to show you here is just this idea of going from gene to protein. We know that there's there are quite a lot of genes that are involved in regulation and things like that. We're just not really going to talk about them as much right now. So we're going to look at this sort of a little bit of a simplistic view of what happens here, because we're just looking at going from genes to protein. Okay, so use of information in DNA to direct the production of a particular protein is called gene expression. Okay, so that's what's meant by that phrase. Genetic information coded in DNA is transcribed. So this is an overview of the process. The first step is called transcription, in which we go from DNA to RNA. And this is a special kind of RNA called messenger RNA, which you may have heard of before. So this whole process of transcription occurs within the nucleus. Okay. So the DNA stays within the nucleus, it never leaves. The RNA leaves through these nuclear pores, which we've talked about. So remember that nucleus is a double membrane structure. The messenger RNA comes out, it's now in the cytoplasm. And then the next stage is what's called translation. And that's where you take the code, which is in the messenger RNA. And the messenger RNA, of course, is complementary to the strand of the DNA that was used as this template. And now this strand of messenger RNA is then used to make protein. So that the process of making protein using the information in the messenger RNA is called translation. And what happens in the cytoplasm is the protein actually starts to immediately obtaining its three-dimensional structure. Why is that? Because of the things we talked about before, right? We talked about hydrogen bonds, ionic bonds. There may be some disulfide links, which are covalent bonds that arise. So so when we talk about a protein, we're talking about a chain of amino acids that has a very distinct three-dimensional shape. Okay. Now they could end up directly in the cytoplasm. So we have free-floating ribosomes. What was the other kind of ribosome we talked about? You guys remember? Some are associated with an organelle. What's that? So you have some ribosomes in your cells that are just floating around, right? So they make protein that your cells directly use, right? What about the ones that are making something like a digestive enzyme that you're going to release into your saliva or something like that? Where are those proteins? Go ahead. Yeah. Yeah, rough endoplasmic reticulum, right? So in that case, what happens is the protein ends up in the lumen of the rough endoplasmic reticulum. It's then packaged up into what are called transport vesicles and then moved to the Golgi apparatus, right? So we talked about that before. Okay. So this whole process of going from DNA to our messenger RNA via transcription, from messenger RNA to protein via translation, this sort of one-way process here is referred to as the central dogma of molecular biology. Okay. Now, again, this is the thing we're going to be talking about today. We know now there's lots of information that goes back to the DNA, right? So we have to regulate expression of genes. So there are proteins and things that will come back into the nucleus and that will have an influence on which genes are expressed and so on and so forth. We're just really not going to talk about that, but that's very important also. The entire process relies on different types of RNA. So we've got RNA, of course, is ribonucleic acid. So we've got messenger RNA, which I mentioned here. We're going to talk about ribosomal RNA, right? This makes up ribosomes together with protein. We'll make up ribosomes. We're going to talk about that later. And then there's yet another kind of RNA called transfer RNA. Okay, so we're going to talk about all three of those today. So let's first make sure we understand what RNA is. So we'll use DNA as our reference. So we all know we have DNA. It's a fairly stable molecule. Why is it stable? Because it's got this double helix structure. In fact, DNA is so stable that as our molecular biology techniques get better and better, they can take an extinct animal like a woolly mammoth, and they can actually extract DNA from it. They can actually find bones of a Neanderthal that died, 100,000 years ago or something like that. And they can extract DNA from it, and they can sequence that DNA. So DNA, under the right conditions of temperature and so forth, and humidity and so forth, can actually be very stable, particularly if it's encased in bone or something like that. RNA is different. It's usually single-stranded, and because of this, it's much less stable. Okay. So if we think about viruses, does anyone know? Can you think of how there's a difference in stability between DNA and RNA and how that affects viruses? Go ahead. Mutations, right? Yeah. So DNA actually, we have mechanisms of copying DNA that are actually really good. Hardly ever making an error, but RNA is much less stable. So there are different types of viruses. Some are DNA viruses where they keep their genetic code is in the form of DNA. Other viruses are in the form of RNA. They're much less stable, and they mutate much more quickly. So the virus that underlies COVID-19, for example, it's a RNA virus, so its rate of mutation is extremely high. Okay, so there's lots of RNA viruses. They mutate very quickly. Okay, so it's less stable. It's typically single-stranded. Now, DNA and RNA do have some similarities. They're both built from nucleotides. They're both nucleic acids, and the basic structural unit is this nucleotide, which we've talked about before. We have a 5-carbon sugar phosphate group, which is PO4, and then something that's basic, meaning it can accept hydrogen ions from the solutions, and it has a lot of nitrogen combined with carbon, so it's a nitrogenous base. So let's look at the nitrogenous bases in DNA. You're familiar with this: cytosine, C, guanine, G, adenine, and then thymine, right? So you need to know those. So then if we look at RNA, we have all the same ones, except instead of thymine, we have uracil. Okay, so I mentioned that earlier. They're very similar, but there's slight difference in chemical structure between the two. And then in terms of the sugar, if you look at RNA, the sugar is ribose. And then for DNA, it's deoxyribose. And this means that an oxygen atom has been removed. So D is removal, so it's like dehydration, you remove water, right? So the deoxyribose, they remove one oxygen. And the number of strands here. So RNA is actually really interesting because we think that DNA, well, there's a little bit of a debate over how our life really started. Was the original genetic code stored with DNA and RNA? And so a lot of researchers, and I mean, it's not 100%, we don't really know for sure, but we think that life may have started with RNA, because RNA not only is it's a little bit simpler being single-stranded, but it actually can fold back on itself and form interesting structures. And we're talking about that. Like, for example, when we look at transfer RNA, that's an example of RNA that's folded back on itself in an interesting way, and it makes a little teeny molecular machine that can do something. So we'll talk about that. So the idea is that in early life might have relied almost exclusively on RNA. And then later on, we developed DNA as a way of storing genetic information much more stably. All right. Oh, so here's some pictures. So here we have ribose. This group, does anyone know the name of this? Have you taken chemistry? What's an OH group? Does anyone know? Hydroxyl, right? So OH by itself in the same water is just hydroxide. When it's when it's a functional group on a molecule, it's a hydroxyl group. So you can see that you have a hydroxyl group at position carbon number 2 in ribose. In deoxyribose, that oxygen is missing, hence the name deoxyribose, missing an oxygen. Okay. Right. Okay. So again, I mentioned these processes before, but the first one is transcription. It takes place in the nucleus. A portion of the DNA serves as a template for messenger RNA formation. We'll talk about that in a second. More detail. Translation takes place in the cytoplasm. The sequence of messenger RNA bases that are complementary to those in the DNA template determines the sequence of amino acids in a polypeptide. So we know about the amino acids. There's 20 amino acids that make up all the proteins in you. The same 20 amino acids make up all the proteins in a plant. Life is pretty much decided on these 20 amino acids. There's actually more than 20 amino acids, but for some reason we use pretty much exclusive these 20 amino acids in almost all of life. And then we have this term polypeptide. So a peptide refers to anything in which you have a peptide bond, which means you have at least 2 amino acids side by side that are covalently bonded to each other. Poly, what does the prefix poly mean? Many, yeah. So a polypeptide is simply many amino acids with these peptide links between them. Okay. tRNA or transfer RNA, the system bringing amino acids to the ribosome. We're going to talk about how that works in a second. Yeah. So here we've got the genetic language of a gene is written in the linear sequence of nucleotides, A, T, G, C bases. The typical gene consists of thousands of nucleotides in a specific DNA sequence. So here, for example, we have gene 1, 2, and 3. It's all linearly laid out. Our chromosomes are linear, as you remember, right? Unlike in DNA, you have a big loop of the main chromosome is just a single loop essentially for bacteria. All right, so that's a DNA molecule. So if we zoom in on this, we can look at our DNA strand, and we imagine you have some super high powered microscope, you actually see these nitrogenous bases, adenine, 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 cytosine, cytosine, guanine, guanine, and so forth. And what happens is during the process of transcription is we make a complementary strand of RNA, which is the messenger RNA. So for complementing A, normally within DNA it would be thymine, right? But we don't have thymine within RNA, so it has to be uracil. So here we have adenine, adenine, adenine. So here's uracil, uracil, uracil. For cytosine, it has to be guanine, right? We talked about that last time. Here we have guanine, has to be cytosine. Cytosine has to be guanine. And then here we have a bunch of adenine, so it has to be uracil here. If we had thymine up here, it would be adenine down here. So adenine is also found in messenger RNA. And then, so that's going to be your messenger RNA. Then we have the next process, which is we want to make a polypeptide, right? So a chain of amino acids. Here's a chain of amino acids that's forming. And the way we do this is we basically look at this code in the messenger RNA, and we look at it in groups of 3 base pairs. So for example, here we have uracil, uracil, uracil here. That's going to be a codon for this particular amino acid, which is just, represented abstractly as a square. Here, the next codon, which is going to be it's called a triplet sequence here, code here. We have G, C, GGC, that's going to code for another amino acid, just represented as a circle here. Then we have CGU, yet another amino acid. And then finally, okay, here we have UUU, uracil, uracil, uracil. Well, we know that that codes for square, so we're back to having a square here. So we can see what's going to happen is we're going to have this big long sequence of amino acids, and that'll make our protein. Okay. All right, so we have a gene, which is a segment of DNA, serves as a template to produce the messenger RNA molecule. The messenger RNA serves to carry genetic information from DNA to the ribosomes, where we're going to do the protein synthesis. Transcription begins when the enzyme RNA polymerase binds to transcription start site on the DNA, known as the promoter. Okay, so there are specific sequences in DNA. Some have very specific functions, right? So there's parts of the DNA that will code for the actual protein. Then you have these other bits that regulate the gene. So, and there's lots of them, right? But here's a very important one, which is the start site. That's referred to as the promoter within a gene. That's a very specific DNA sequence. Okay, the RNA polymerase joins new RNA nucleotides in a sequence complementary to the DNA to form messenger RNA. Messenger RNA has a sequence of bases complementary to the DNA, except we're replacing, instead of thymine, we have to have uracil. I mentioned that earlier. And then messenger RNA is a faithful copy of the sequence of bases in DNA. All right, so let's go through this. I think I already mentioned most of this, but the translation phase is the second process with which genes expression leads to protein synthesis. Translation occurs at the ribosome, requires several enzymes and different types of RNA molecules, messenger RNA, transfer RNA, ribosomal RNA. There's 64 different messenger RNA codons. So where does this number 64 come from? Does anyone? So it's basically the idea that you have 4 different possibilities, and then you have 3, right? So let me see. 4, let's do the math together. I didn't actually do the math. So if you have 4 different possibilities and 3 different, essentially, letters, right? I think it's 4 raised to the 3rd power. Is anyone good at math? 4 raised to the 2nd power is 16, right? And if you raise that again, that doesn't quite work, does it? Maybe it's, yeah, it's not right. How do we get to the 64? It's 4 raised to the power 3? Okay, 4 times 4 times 4. Yeah, that's 64? Okay, so, okay, that's right. All right, sorry. It's too early in the morning, right? So 4 raised to the power 3 is 64. So this is just a basic, this number 64 is nothing magical, right? It's 4 times 4 is 16. Multiply 16 by 4. Yes, of course, it's 64, right? Sorry, it's early in the morning, right? So that's just the consequence of math, right? Where that's, of course, you have to have 64 different possibilities if essentially you have 4 letters, right? Okay, then you have them in, three letters, one, AAA. If you go through, it'll take a long time, but you could go through all the possibilities. You actually see a chart next, right? We'll have to see all 64 possibilities. All right, so that's where we get that number from. Sorry, it's a little early in the morning for math for me. So each nucleotide unit or triplet of messenger RNA molecule is called a codon. Now we have 61 different codons. So this is kind of an interesting fact. So these are 64 possibilities, right? Just by math, we have to have that. But there are 64 actual codons that will code for a protein. Code for protein. So that means there's some others that are not going to code for protein. But we're going to get to that. So what are they doing? We'll see what they're going to. It's a little bit of mystery. Oh, here's the other 3. They're down here. The remaining 3, right? 61 plus 3 is 64, are what are called stop codes. So we have 3 codons that are stop codes. So as you're making protein, you need to start. So we'll talk about how we start the process. But the ribosomes also need to know when to stop the process, right? So there's going to be certain stop codons. So uracil, adenine, adenine, uracil, guanine, adenine, uracil, adenine, guanine. Those are the 3 that are going to signal determination that you essentially finish the protein. Okay. Let's take a look at this chart. And I know it looks confusing, but it's actually not that bad. So here's all 64 possibilities. So one interesting thing is it says here the genetic code is almost universal among all living organisms. So this is kind of crazy, but say your cell wants to make phenylalanine, then the codon for that would be uracil, 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 or it might be uracil. There's two possibilities: uracil, uracil, cytosine, right? What if a tree wants to make phenylalanine? What code does it use? It does the exact same thing. What about bacterial cell or yeast cell? They use the same thing. So there are occasionally slight differences. There are, and those do exist. And things that are really distant from humans, like if we're getting, so a tree would be exactly the same as us, pretty much. They're also eukaryotic organisms. As you get further and further away from this, there might be some very small differences here and there. But in general, almost all life uses this exact same code, which is kind of crazy. What does that imply if we're all using the same code? Well, it's thought that every single organism on Earth, including bacteria, archaea, and so forth, all arose from one organism. So life arose once, and that one initial organism, whatever it was, some sort of single-celled organism, just mutated and divided and changed over time, and now we have all the incredible diversity of life. So essentially, if you're eating, let's say you eat a salad, you're essentially eating your cousin because it's a distant relative of you, right? All right, so we all use the same code, which is kind of amazing. Each triplet codon codes for a specific amino acid. So if you look at anything like this, like what does that mean? So if you say uracil, uracil, adenine, that's always going to make leucine. Okay, so in that direction, it's always going to make leucine. But sometimes there are multiple amino acids. But for some amino acids, there are more than one codon. So it goes the other way. Here, like leucine can also be, here's another codon, U. So you've got UUA and you've got UUG. Both of those codons will both make leucine, okay? So it's possible to have multiple codons and make a single amino acid. However, for a given codon, you can only make one thing, right? One amino acid, okay? Okay, how do we start the process of coding? Oh, let's look at how this, do people understand how this chart works? It's a little bit weird in the beginning, but you have a triplet code, right? So the first base pair, let's say you wanted to look up, let's look up UCA. I'm just throwing something out of the, out there. So you say, okay, the first base, nucleotide is U, uracil. C, you go up to the top, you see, okay, you go to the second column, so we're up here in this row, second column, it's C, and then I said A, right? So then for the third base pair, you go over here, and you read down here, here's the A. So UCA is here. Okay, you see how that works? You look first over here, then to the top, then to the side, and you can quickly find whatever you want. Okay, so serine is going to be the amino acid that's made there. Okay, that's how this chart works. Now, let's say we need to start production of a protein. How's it occurring? Well, it's always going to be start with this codon AUG. So let's look up AUG. So look up, here's A in this row. We look to U up here in the column, and then G. So here's U, and then we look over here for G. A, U, and then we got G, and then that brings us to methionine. Okay, that is a codon that starts every single protein in your body, forming the same thing in the tree, same thing in a yeast cell. So that's the very first amino acid of any protein sequence. Has to be methionine. Okay, and then how do we stop the process? Well, we have these codons. They're actually kind of grouped together, UAA, UGA, and UAG. All these are the stop codons. So they do not code for amino acid. They just tell the ribosome to stop making proteins. Okay. All right. So let's look at some of the other players here. We need to understand what transfer RNA is. So this is, as it says here, it acts as a molecular interpreter. It carries amino acids. So it's like a little shuttle that carries things around. as you can see here, the amino acids bind to one end. So there's a binding site for an amino acid here. Here we have phenylalanine. The transfer RNA is an example of a single-stranded RNA, but it bends back on itself, forming these bonds. Okay, so it makes this little molecular machine. And at the other end, the polar opposite of the side where you can bind to an amino acid, you have a region. For example, here we have adenine, adenine, adenine. So, and this is called an anticodon. So it's a sequence of three bases, nitrogenous bases, and this can actually form a complementary interaction with a codon, right? So here, A and U, of course, can and hydrogen bond to each other, right? So the amino acids bind to one end, the opposite end has an anticodon, which matches its complementary codon in the messenger RNA. So you're going to have many different transfer RNAs, right? Each with a different anticodon. That means it can bind, each of these anticodons can bind to a different codon. So codons and anticodons both have these three nucleotides, right? And then the order of the messenger RNA codons determines the order in which transfer RNA brings in the order in which transfer. So it's gonna, so for example, this one here, this anticodon here, that's always going to be that's going to be able to hydrogen bond with this particular codon. You'll always have this particular amino acid, right? So if you have a different codon, a different transfer RNA with a different anticodon, you'll have a different amino acid. Okay, so let's go back to ribosomes. We've talked about ribosomes before. Ribosomes have a lot of protein, but they also have ribosomal RNA. So they're a little bit of a mixture of protein and ribosomal. So rRNA. And there are two subunits to ribosomes. There's a small subunit and a large subunit. So where do we form these subunits? Does anyone remember? Where in your cells do we actually start to put together these subunits? Where do we form the subunits? Go ahead. Well, I could guess. No, it's there. So RNA is a little special. It's actually, remember there's a dark area within the nucleus? It's called the nucleolus, right? You can actually see that sometimes with a light microscope if the cell is stained properly. So you can see this darker area. That's where you have the ribosomal subunits being manufactured essentially within the cell. Okay. Now they don't come out as a complete ribosome. They actually come out as the two subunits, large and small. Each will come out individually through the nucleopore, and then they're actually going to be that final step with some assembly we're going to talk about actually occurred in the cytoplasm. Okay. And so that might be what you're thinking about, right? So we'll see that in a second. But that's what I was talking about is the earlier, where do you actually make the ribosomal subunits that actually occurs in the nucleolus. Okay, so subunits join in the cytoplasm just before the translation is to occur. We'll talk about that. So this ribosomal RNA, it has to interact with the messenger RNA, so it has to have a binding site for that. And it's also going to have to interact with the transfer RNA. So we'll have binding sites for the transfer RNA. So these binding sites facilitate complementary base pairing between the transfer RNA anticodons and the messenger RNA codons. We'll see a picture of that in a second. The polypeptide forms, the polypeptide, again, remember what is, that's a sequence of amino acids. It forms as new transfer RNA molecules arrive. And then translation terminates at the mRNA stop codon. And we'll see, this will all be clear when we look at a picture of this. So here's a nice picture. And we're going to go in more detail, but this gives you a good overview. So here's what it looks like. A ribosome looks like. You have a small subunit and then a large subunit. What's going to happen is it has a binding site for the messenger RNA. So the messenger RNA sort of fits in a groove within these small and large subunits. And then we have these little slots, which are binding sites for the transfer RNA. And they're labeled APE, okay? So we'll talk about that in a second. So what can happen actually is when and this happens in both bacterial cells as well as in eukaryotic cells, as you have a strand of messenger RNA, it can be very long. what you can have is multiple proteins being made at the same time. The way that works is the ribosomal RNA will essentially glom onto and we'll talk about how this occurs in a second. It'll attach itself to the messenger RNA, and it'll actually start moving along as it's reading. So again, this messenger RNA is a code, it has a code on it, right? So with the ribosome will actually move along this, and as it's moving, it'll start to generate a protein, which is a polypeptide here. And as it moves along, this polypeptide will get longer and longer and longer. But what can happen is your cell doesn't just wait till the one protein is done. As soon as one ribosome moves a little bit of a distance along the messenger RNA, it's starting to make the protein, another one will come and they'll start making a protein. And yet another one, and yet another one, and so you have one strand of messenger RNA and many, many ribosomes all reading the information and making proteins, okay? So this thing is called a polyribosome. Poly just means many again, so it means you have many ribosomes all attached to a single strand of messenger RNA, and each one is making its individual protein, okay? All right, so let's look at this overview, and then it's going to go pretty quickly. We'll take a look at this, and then we'll look at all the steps in a little bit of detail. No sound. No sound. Okay. Let's stop this. Oh. It's all the way up. Huh. Input. Direct.
How is the info?
Right. Completely frozen. I can't even get out of it. Oh. Yeah.
How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are three distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down one codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation.
Okay, so that's it. So that we're going to go through all the steps that were shown there.
How is the information in the
Okay, stop. All right, so let's look at all the steps that they mentioned. I think that was a really very nice, concise video, but let's look at it in a little more detail here. So we've got, how do we actually start the process? So we look, this is called the initiation of protein synthesis, the large. So we're going to start here. We have this small ribosomal subunit binding to the messenger RNA. And then nothing can happen until an initiator transfer RNA pairs with this messenger RNA. And it's always, again, going to have this, we're looking for this code AUG on the messenger RNA, that's the codon, the anticodon be UAC. So this is a particular transfer RNA that can bind there. And it's going to have methionine attached to it. So it always starts that way. What's this complex? A complex is when things bind to each other. So this is now a little complex here. Once this complex forms, then the large ribosomal subunit can come in, and it forms an even bigger complex. And now we've got the beginning of the actual synthesis can happen now. So we have, again, these three sites: the A site, the P site, and the E site. The E site just stands for exit site. Oh, here they have the names here. So E stands for exit. That makes sense. So the transfer RNA will come out over there. The A site stands for amino acyl, and then P is sort of peptidal. You don't really have to know that. Just remember A, P, and then E. Okay, so the transfer RNA will come in here for the A site. Okay, so you can see the three binding sites located directly adjacent to exposed messenger RNA. It's the RNA transfer RNA molecules that bring amino acids to the ribosome to use for making proteins. So the larger, here it says the largest ribosomal unit completes the ribosome. Initiator T RNA occupies the P site. So initially we have something in the P site. And the A site is now ready to be occupied by a new transfer RNA. So, okay, so we have something starting here again in the P site. This A site is unoccupied. So here, for example, we have transfer RNA with a particular amino acid. I think it's aspartate, but it comes in with the anticodon. It's going to there'll be complementary base pairing. Then what we do is we form a peptide bond. Remember I mentioned a peptide bond that's between amino acids that forms with the covalent bond. It's pretty strong. So that's occurred here. And then the next thing is this entire ribosomal complex that moves down essentially one codon, right? And so the video mentioned that this transfer RNA, it's uncharged, meaning it no longer has an amino acid, so it then leaves. This thing that was, which was originally at the A position, now ends up is now in the C position because the whole ribosomal unit subunit moved downwards. We have an unoccupied A site and now a new transfer RNA with its associated amino acid can come in and occupy the space, and then the process continues over and over again. Go ahead.
So it always starts in a P site?
Yeah, it always starts in the P site. So if we go back there, it's a good question. So this is the sequence for every single organism pretty much on Earth. It's going to start this way, where you have the messenger RNA, small ribosomal subunit, then transfer RNA with a methionine, and then the big ribosomal subunit comes and it gloms onto the whole thing, right? So this always ends up in the P site, and you always have methionine as the very first amino acid for every single protein in your body that your cells make. Okay, and that's convenient because now this A site is unoccupied, and then a transfer RNA can come in with the next, which it'll have the anticodon that goes with this codon, and then you'll be able to make the next peptide bond, right? And then this will get longer and longer and longer. So the ribosome is essentially moving down, reading the messenger RNA, and then you have this nice string of amino acids coming off. And as I said, the amino acids, because they'll form hydrogen bonds and things like that, it'll actually start to curl into whatever structure it'll have. You might have remember with the alpha helix, you might see an alpha helix starting to form. You could potentially see a beta pleated sheet, which is bends back on itself, right? So those things start to form right away. Okay. So termination, how do we terminate? Okay, so imagine we've made maybe 300 amino acids. So it happens to be a protein with 300 amino acids. Well, we don't want to just keep continuing to make the protein, right? We got to stop at some point. So there'll be the stop codon. Here's one UGA, one with the 3 different stop codons. A release factor will come in. It does not have, it's not a transfer RNA. It does not have any amino acids, so no peptide bonds can form. But this essentially is a signal that, okay, we need to break this whole thing apart. And now we have our protein, all the subunits are separated, this transfer RNA is separated, the small, large subunits, and the messenger RNA, they're all sort of disassociated. So the whole complex is now falling apart. So termination is the final step in protein synthesis. The termination of the polypeptide synthesis occurs when the ribosome comes to a stop codon. A release factor binds to the A site, where the stop codon is exposed and releases the polypeptide. The polypeptide is free to take on its 3-dimensional shape. It's like I said, it's probably already started to form it, but now it's really completely free, right? So now it really makes its final adjustments into its final 3-dimensional shape. The ribosome dissociates into the 2 subunits, but then again, the whole process can start again, right? So the small subunit, it can go back to the start codon, attach glom on there, attach there. The transfer RNA with methionine would come in, occupy that, and then the large ribosomal subunit would attach itself, and then the whole process starts again. Okay. So, any questions on that? It's pretty confusing. Is it okay, or go ahead?
So it shapes the 3D form after breaking off or before?
Well, it actually does both. It starts. If it's a long enough protein, a lot of proteins are very long, it's not going to wait. It'll actually start doing some of those. Okay, let's go back to what we know about proteins. There's the primary structure, which is the sequence of amino acids, right? The secondary structure is usually a helix, an alpha helix, which is a coil. The other kind is a beta pleated sheet. What does a beta pleated sheet look like? It basically looks like this. I'll show.
Like a ladder?
Yeah, it kind of looks like, yeah, it goes back and forth a bit like that. Like that might be a beta pleated sheet. Then you might go on and have an alpha helix. And then maybe you'll have a very small beta pleated sheet right there. I mean, it could just bend back on itself like this. Makes this kind of what's called a hairpin turn, goes like that. Maybe it'll make another alpha helix. So it'll actually start to do that right away. But I think the point that they're making here is that as long as you still have one end attached, it's probably going to inhibit it from fully making its 3-dimensional shape, right? But once this is released from the messenger RNA and the ribosomal RNA and all that, now it can sort of spring into its final shape. Okay? Yeah. All right, that's a good question though. So, all right, so here's our overview. DNA. It's the first step. It's the template. It's going to serve as a template for messenger RNA. That's called transcription. And we're actually going to make. There's something I want to mention here, which has to do with which I didn't mention earlier, but it's in this figure. So let's go ahead and tackle it. Here's our primary messenger RNA sequence. Let's go ahead and draw it. So you got this linear piece of code essentially. Now you actually have sequences, bits in here that are called introns. How did they do it? Oh, I'll do it the way they did it. So look at this. Here's the intron. Here's another intron. Okay. So I've drawn 3 introns here, okay? Now, intron. It comes, the int part comes from intra, which means within this piece of DNA. I forget what the tron mean, man. I'd have to look it up again. But there are these little bits here. These introns are here. And they're essentially, they're genetic material, but they're not useful for making the protein. So they have to be excised. So within the nucleus of your cells, you have special enzymes that will chop out these introns. Okay. So if these are called introns, what might you guess the other bits are called? Does anyone remember? Or do you have it in biology or no? Exons. Yes, the other parts are called exons. Is that right? My spelling's not the best. I think they're called exons like that. So these are the bits. Now, exon, the name's a little bit, it comes from expressed, right? The X comes from expressed. I have to look up what the on bit. So this comes from the word expressed. So these are the bits that are expressed that are actually going to code for a protein, right? So what you have is, once you remove the introns, you'll have a shorter bit of genetic material. Well, not genetic material, but of code. This is messenger RNA, right? It has a code on it. It's shorter. And that's the thing that's actually going to move. That's the mature messenger RNA. You have enzymes that have removed all the introns. And then this thing can exit through the nuclear pore. Once it's outside into the cytoplasm, we can go through the whole process. Here we have, so step 2 is processing the messenger RNA, removing the introns. 3, we have messenger RNA moves into the cytoplasm, becomes associated with ribosomes. We just discussed that. 4, transfer RNAs with anticodons carry amino acids to the messenger RNA. 5, where the anticodon complementarity base pairing occurs. And that allows us to make a very specific sequence of amino acids, which will form our peptide, which will become the protein. So the polypeptide synthesis takes place 1 amino acid at a time. Okay, so. All right, so let's look at how this occurs and compare prokaryotes to eukaryotes. So here we have a prokaryote, which could be most likely a bacterial cell. We have RNA polymerase that makes the messenger RNA. Remember that in a prokaryote, there is no nucleus, right? So what happens is the messenger RNA is starting to be made, come out, and right away, immediately, the ribosomes will attach there. Again, you have large and small subunits. Ribosomes are actually slightly different in prokaryotes compared to eukaryotes. They're slightly smaller, they're not quite as complex, but they essentially perform the same function. And you can see this is a polyribosome, multiple ribosomes all working on the same piece of messenger RNA. Oh, they're moving in this direction, excuse me. So this is the 5' end, this is the 3' end of the messenger RNA. So it's moving along this way. And in fact, you could have multiple ones all working on the same piece of messenger RNA. Okay. So that's a polyribosome there. Let's look at a eukaryote. So this would be something like your cell. So we have the DNA. It opens up. Again, we have RNA polymerase. It's going to make a piece of messenger RNA. Note that this piece of messenger RNA has some introns, which are shown in gray. We have special enzymes that will sort of make little loops and then excise these introns. And now we just have the final or mature piece of messenger RNA. This can leave to the nuclear pore. And then we have ribosomes that will, starting over here, they'll move along this messenger RNA. And you can have multiple ones. Again, you have a polyribosome. And they're all coding, essentially converting the code from messenger RNA into an amino acid sequence, which is then a polypeptide. And a large polypeptide is considered to be a protein. Okay, so in prokaryotes, transcription and translation both occur in the cytoplasm. In eukaryotes, transcription occurs in the nucleus, and translation always occurs in the cytoplasm. We're going to go on to a different topic, but let me just talk about one thing that may be a little bit confusing because I've used the terms polypeptide and protein on kind of almost synonymously. So let's go back just for a second on this terminology. So the term peptide is used a lot in biology, right? So a peptide are amino acids, at least two, usually a lot more, could be like 8 or something like that, or even hundreds, that are bound together with a peptide bond. Okay, that's what a peptide is. Polypeptide pretty much means the same thing. It's poly often means a few or many. So maybe if it was 2 amino acids, you wouldn't use the term polypeptide. It'd have to be like 3 or 4, right? So again, are there exact numbers? No. Like these are just sort of the general sense is poly might be, 4, 5, 6, 7, 8, or even hundreds, right? So all proteins are technically polypeptides. So why don't we just use the word polypeptide all the time? Why do we have the term protein? The term protein is actually very useful because once a polypeptide reaches, again, there's no hard set rule, okay? But roughly around 100 amino acids, it starts to have an actual 3-dimensional shape that's quite interesting, like a globular shape or a fiber shape. That's around the tipping point where you start to call this thing a protein because it has a very interesting 3-dimensional structure that's involved in its function, okay? So again, if you look in the textbook, someone will say, well, exactly from 99 to 100, we switched from using the word polypeptide to protein. That's not the way it's really used in science, okay? That's textbook ease, trying to make things easier for you. But a protein is generally a very large polypeptide, but it is a polypeptide, and the term polypeptide is sort of encompasses all these different things, except the very smallest, like 2 amino acids might not be considered polypeptide. All right, sorry, but that's confusing, and it comes up over and over again. People are confused. Why do we have so many terms for the same thing? All right, yeah, go ahead.
Before you go on, put this in context. As I understand it, each strand of DNA is going through transcription and translation.
Right.
How often does that go on?
All the time.
All the time.
Well, okay. Let me qualify that a little bit. So you have skin cells, you have other cells, but you have different types of cells in your body, right? So for example, a skin cell does not need to make a digestive enzyme the way a cell in your pancreas would, or a cell in your salivary glands in your mouth, right? So a skin cell would essentially never turn on the genes for making a digestive enzyme, right? Like amylase is a digestive enzyme that breaks down starch, which is found in corn and potatoes and things like that. Why would your skin cell ever do that? So it will never turn on that enzyme. It will never turn on it has the gene, right? But it will never turn it on and use it to make protein, which would be the digestive enzyme, because there's no point, right? So, and likewise, they're like a cell within your salivary glands or within your pancreas, they're making digestive enzymes, things like that. They don't need to make keratin, which your skin cell has, which is the hard waterproof coating you have. This really tough protein is called keratin, right? It's also found in hair and nails. They don't have to make that, right? They would never turn that gene on. So there are certain things that will never get turned on because the cell has gone down. Its fate has now become this, right? It's kind of like life. Like people make choices about their career and then they end up specializing, right? So there are certain genes that will never get turned on. Okay. Other genes are more like housekeeping genes that the cell needs just to stay alive. So every single cell in your body, like glycolysis is a very important process for turning glucose into pyruvate and getting energy out of it. And it's the beginning steps that leads us to making lots of adenosine triphosphate, which we use for energy, right? So every single cell needs that in your body. So every single cell will be making those sorts of enzymes. So they'll be turned on all the time, and they'll be continuously making these certain enzymes and so forth, right?
So there's a strand of DNA in every cell.
Yeah,
There's 35 trillion cells in the body.
right.
That's a lot of stuff going on.
So, yeah, I'm glad you noticed that. So basically what's happening is, it's absolutely incredible if you think about it, which is that your body is constantly working for you all the time, whether you appreciate it or not. You should probably spend a moment every day of just appreciating what your body's doing for you, what all these molecules, what all these cells are doing, all the molecules are doing for you every day to keep you alive. It's astounding. Go ahead.
So is some people's cells always working, like with nails in here? Some people hair grows.
Yeah, yeah.
More.
Well, so that has to do with age. So as people age, these metabolism decreases,
Mhm.
and so the growth of nails and hair and things like that gets slower. But yeah, and there's differences between different people. But yeah. All right, so let's look on. So now we're switching over to a different topic, but it's a very interesting one, and it has to do with a lot with life and with lots of different aspects of life as well as with health. So this topic is gene mutation. So what is a gene mutation? It's a permanent change in the sequence of bases in DNA. So what can a gene mutation do? Well, it can do both good and bad things, right? So effects can range from changing expression of a gene to completely inactivity of protein. So we'll talk about specific examples of this, but you could have, well, I'll give an example right here, and we'll come back to it, we'll circle around to it. I'm going to use the example of sickle cell anemia. So that's going to be a single change in a single protein within hemoglobin, which is a protein that we use to carry oxygen, and it's found within red blood cells. So it's a change in just a single protein changes the whole shape of the protein, and it tends to clump together, and that then leads to this sickle cell shape of the red blood cell, right? So a very small, what looks like a very small change can have a really massive impact on the final protein product. But in that case, the hemoglobin protein is made. People with sickle cell anemia, particularly if you're heterozygous, actually are perfectly fine for the most part, right? In fact, they're essentially immune to malaria. However, there are also changes where you have a change in a gene, and now the protein is completely inoperable. It's completely useless, right? So you're just simply missing that protein from your whole body. And there are examples of people that are simply missing certain proteins, and it's kind of sometimes amazes medical science, because they say, well, you're missing this protein, and yet you're still alive. And we thought this protein was essential to life. So, but that can happen. You can just completely be missing a protein because it's completely malformed, it doesn't work at all. All right, so where can genes occur? Well, they can occur in what's called the germline. What is the germline? These are cells that will make your gametes, right? So for a male, they would make the sperm, and a female, they would make the eggs. So if you have a germline mutation, that can then be passed on to the next generation, and it can pass on from that generation to the next, to the next, to the next. So what's an example of a germline mutation? A classic one is, we know that humans all came out of Africa, and humans, with almost 100% certainty, we know that all humans had brown eyes, right? So we know now there are people that have green eyes, blue eyes, and so forth, right? So that was almost certainly a mutation in the germline, right? So if you have blue eyes, you're, in quotes, a mutant. Now we don't call people that have blue eyes mutants, right? Because now the more polite term is it's been incorporated into the population. Plenty of people have blue eyes. It's a genetic polymorphism, but the origin of that was in fact a mutation, right? So again, it went into the germline. It was passed on from generation to generation. Okay. So some germline mutations can be responsible for susceptibility to cancer. Some will make your eyes blue and so forth. So there's a lots of variations, right? So we can talk about that more in a second as we get to, we'll talk about cancer later. Now, there's other possibilities of somatic mutations. So soma, the term soma just means body. So somatic mutations in your body cells that are not, essentially all the cells in your body that are not germline are somatic cells. So if you have a mutation in a somatic cell, these cannot be passed on to the next generation. So for example, if I went out without sunscreen and I got a little spot on my forearm here, and it turns out it's cancerous, right? That's kind of scary, but that would be a somatic mutation, but there's no way I can pass that on to the next generation, right? Because the mutation didn't occur in my germline or in sperm cells or anything like that. So that's a somatic mutation. It can't be passed on. It can lead to cancer development. Finally, we have this weird thing at the bottom that says mutations can be good or bad. So mutations can lead to things like potentially cancer. It can increase your susceptibility to cancer. It could lead to your cells in your body making proteins that are completely inactive, that don't work at all, right? That all sounds very negative. So can anyone think of why mutations might be good? It sounds actually tragic almost, like, okay, go ahead.
Like adaptability
Yeah.
when environment changes.
Sure, that's a good one. You have one? Yeah.
Can't certain genes cancel out other genes?
Sometimes, yeah, it's possible, yeah. So you're saying if you had a maladaptive mutation, you might have some other genes that compensate or something? That's
Yeah,
definitely possible.
yeah.
Yeah, so that actually happens a lot. Like I was saying, like I said a second ago, like sometimes people are missing a gene because it's completely inoperable. And the scientists are like, well, how can you be alive? And it turns out they're like a very critical thing for a biochemical pathway in a cell. But it turns out that the cells, every single cell in their body found an alternative pathway to make the products. So, well, that would be more of a neutral mutation, but it wouldn't. So, but okay. So mutations clearly can be bad, and they can be good. So as Chloe said, what can happen is basically mutations have led to evolution, right? So we wouldn't have evolution without mutations. So as I said, every single organism, as far as we know, came from a single living organism. And it would have been very boring if for billions of years, Earth just had one organism, right? So when you go outside and you see there's birds, there's mice, there's chipmunks, there's all these different organisms, and jellyfish, all these really crazy things. Well, they're all your distant cousins, and they're all here because of mutations. So we can, all the diversity of life, which is amazing, really, right? Think about sperm whales, think of all these different things. They would not exist without mutations. So this had to have happened at some point. Okay, so how do mutations occur? So some occur spontaneously. They arise due to abnormalities in normal biological processes. So it turns out, for example, if you look at within your body, your DNA is essentially always under attack. There are things called ROS, reactive oxygen species. And so these are highly reactive substances involving often oxygen, like hydrogen peroxide we mentioned earlier, right? Those are found in peroxisomes. There's other forms of reactive oxygen species. And just through regular metabolism, as you have a meal and then your cells process that glucose and all that, your mitochondria will make reactive oxygen species. And these are so reactive that they can break your DNA, they can essentially knock nucleotides out and substitute new ones. They can do all sorts of terrible things. So that leads to some of these abnormalities just from normal daily life, just from eating a meal. You're going to have some of this happening. Now, you have a lot of mechanisms like enzymes, for example, in your cell that are fighting the effects of reactive oxygen species. And there's actually something else that you should do daily, which would help you fight the reactive oxygen species. Does anyone know what they are? So reactive oxygen species, that's a little bit of clean oxygen, anti-something. What? Why are you told to antioxidants, right? Why, since time immemorial, have people been telling you to eat fruits and vegetables? Fruits and vegetables contain antioxidants, right? Like vitamin C. Those are incredibly powerful antioxidants. So they help a lot, right? So you should always have your fruit and vegetable every day because they will help fight these natural processes that are happening. But despite that, even if you're having your antioxidants every day, through fruits and vegetables, you're still going to get some of these spontaneous mutations. There's really nothing you can do about it, but you do have repair processes within your cells that will try and repair the DNA. Now, you also have induced mutations. So it's about 50-50, by the way. So about half your mutations that occur through environmental influences. This is things like heavy metals. There's all sorts of horrible things in our, that make their ways into our food and so forth, right? And our water supplies and things like that can induce mutations. So lots of different chemicals. All right, then we can have other things that can happen is just, even without these first two things, the spontaneous mutations, well, this actually leads to these spontaneous and induced mutations or errors in DNA replication. So there's also the ability. So first of all, your DNA, when you have DNA replication, when a mother cell turns into two daughter cells, it has to replicate the DNA. That process is incredibly good. It has almost no errors. So here it says right here, typically 1 error per 1 billion nucleotide pairs replicated. So it doesn't have a lot of errors. We do have mechanisms to try and fix the errors if they do occur. But they still happen, okay? So DNA replication errors do occur. And this can lead to mutations. This is just part of the process of this molecular machining not being perfect, right? Nothing's perfect in the world, right? So proofreading by DNA polymerase usually minimizes errors in your strands, but occasionally mismatched pairs do occur, okay? So that's a rare thing, but it does happen. So these are different sources of mutations. Again, spontaneous mutations because of things like reactive oxygen species, errors in DNA replication process itself, and then induced mutations through environmental influences like heavy metals, all sorts of chemicals. So now we have, we'll talk a little bit about the things that can cause these mutations. So the first thing is a mutagen, right? So the term gen, it's like an electrical generator, right? Or genesis. It's something that gives rise to something. So it creates mutants, right? So what creates mutants? One thing is environmental influences. So environmental influences can cause this. So it's a mutation, again, is a change in DNA sequence of an organism. So here's some examples. So radiation. So in like a lot of the comic books and so forth, this is a favorite thing, right? Scientist gets a huge dose of radiation and now he has superpowers. X-rays, they're actually cosmic rays coming from the sun. They get hit by those. UV rays are also mutagenic. So UV ray, these are essentially just light that has a very short wavelength, but it has a lot of energy because of that. So it can break DNA within your skin cells, for example. Certain organic chemicals, cigarette smoke, certain pesticides are all. So the list goes on. There's hundreds and hundreds of different possible mutagens. So these will cause changes of the DNA sequence of the organism. So DNA repair enzymes constantly monitor repair any irregularities, generally keeping the mutation rate low. So basically, every time you go out and you're exposed to something like UV rays, you're probably damaging your DNA a little bit. But luckily, we have all sorts of repair mechanisms that cells have developed over millions of years. That helps us. Now, there's another term which is carcinogens, right? And this is a little bit confusing. Again, the word generate here, it's going to make cancer, right? So it's going to make cancer. So carcinogen means a substance that will lead to cancer. So what are some of these things? Well, there's a lot of overlap here. Things like cigarette smoke could be a carcinogen. But here's the thing: not all are. Some but not all carcinogens are mutagens. Okay. So how is that possible? Well, the way some carcinogens work is that they're in fact mutagens. So they induce permanent changes in your DNA. However, there are some carcinogens that don't affect your DNA at all. They just, they will induce your cells to become abnormal and replicate without stopping, right? So it's possible to be a carcinogen and not be a mutagen. And let's go back to mutagens, because it sounds like these two terms, there's a lot of overlap between the two terms, but it's actually possible to be a mutagen and not cause cancer. So an example would be, again, imagine I was a mutagen anthropomorphizing here, but I'm a mutagen and I cause you to have a child who has blue eyes. That's fine. They don't have cancer, right? All it did, but we did change their gene, right? We did a permanent change in the gene, but they have a change in eye color, but no cancer, okay?
Well, everything that's in, mutagen, I thought that is cancer too.
No,
That be cancer?
well, in general, as I said, there's a lot of overlap, but we have different terms because a mutagen is not necessarily cancerous. Because again, you could even have a mutagen that causes a mutation that's actually advantageous, right? So it's actually possible that it doesn't cause cancer or do anything, but the vast majority do cause cancer, so right there. Carcinogens, most of them are mutagens because they actually cause genetic changes, but some carcinogens actually act differently. They just stimulate your cells to divide like crazy without actually changing the DNA at all.
Okay,
So-
so too much of a mutagen can cause cancer, basically?
Yeah. So,
Okay.
well, again, mutagen, it just means that you've changed the DNA. Often that leads to cancer,
Okay.
but it doesn't always, right? Okay. It's confusing because the terms seem to all very similar, but they're slightly different. Finally, we're going to get to this last idea here, which is really interesting. So another way of changing your DNA permanently, potentially permanently, are these things called transposons. And they're referred to as movable elements. They're essentially genes that move around. They can jump around from one location to another. And this does occur in humans. It occurs in plants, such as, for example, was first studied in corn. Okay, so it's responsible for causing mutations because the idea is you have a sequence of genes, right? So you have genes that's gene 1, then you have gene 2, gene 3, and so forth, right? But now you have this bit of a transposon, which jumps into, let's imagine it jumps into the middle of gene number 1, right? It's going to completely disrupt that gene, right? And stop it from working most likely. It can actually, and transposons are interesting, they can do like a copy, they can copy themselves over to this and remove this original location, or they can leave themselves in this original location plus in this new location. So the crazy thing is about half your DNA probably, you have a lot of DNA in your cells, about half of it probably arose by the fact these transposons are jumping around, which is completely crazy. So it can cause mutations. Sometimes it doesn't cause any effects that are negative, but often it will cause mutations because it'll just pop itself right into the middle of a gene. So I'm not going to talk a lot about transposons, but I just want to mention this historical figure because she's so important. This is Barbara McClintock, Nobel Prize winner in 1983 from physiology or medicine. So in the 1950s, she became very interested in genetics. She had taken a class as an undergrad, and that set her on the course of work studying science and so forth. And she found that corn was sort of amazing. You probably have all seen this, right? Particularly in the fall, we have this really interesting corn that's really beautiful, right? So she began to study these mosaic patterns of corn or maize genetic level. She noted that the kernel patterns were too unstable and changed too frequently over the course of several generations to be considered mutations. Mutations occur very rarely, okay? So what could be possibly happening to cause all this tremendous variety that was happening in corn? So what was possible is the answer contradicted prevailing genetic theory. So she started publishing a few papers in the 1950s. She continued to work essentially her whole life until she died when she was 90. And she kept working on genetics and various other things. But her ideas in the 1950s were so radical that no one believed her. They were just like, thought she was crazy. And that actually prevented her from publishing probably as many papers as she would have. But she was literally about 30 years. So between the 1950s and when she got her Nobel Prize, which is literally 30 years later, finally science caught up. And what she had found is she actually found these transposons. She went on to discover such mobile elements, now known as transposons. She found that depending on where they were inserted into a chromosome, these mobile elements can reversibly alter the expression of other genes. So anyway, it's a very interesting, we'll end on this topic. She's a very interesting person. And she was so far ahead that no one believed her. But in the end, everyone had to admit that she was absolutely right. And so she's now sort of a legend. All right, we'll stop there.
Begs the question, where we can come up with that idea.
Yeah. You have to wonder, right?
Yeah.